In order for cells to produce proteins, they must first synthesize something called a messenger RNA that is generated based on DNA gene sequences. And as the name implies, these RNAs are intermediate messenger molecules that can carry instructions for producing specific proteins. And on occasion, the message can be modified through the process of alternative splicing, which could potentially alter the final protein product. And my main research area would be the study of alternative splicing, and with the ever-increasing availability of sequencing technologies, we're able to interrogate large numbers of these alternative splicing events by examining the RNA species produced by specific cells under specific conditions. And in my research work, I focus on the impact that RNA splicing has on the maturation of white blood cells, a highly diverse group of cells that play critical roles in immunity, among other functions. And so there are two main reasons for why we're studying this cell type. And firstly, the effects in alternative splicing are especially implicated in some kinds of leukemias, where certain white blood cell types fail to mature, and this implies that RNA splicing may be particularly important for these cells. Secondly, each white blood cell type, and sometimes even subtype, is highly specialized for specific roles, yet they have to remain responsive to a range of potential threats. And alternative splicing could be one of the players in conferring this functional flexibility to these cells. And presently, through RNA sequencing, we have identified several candidate regulatory factors that may be regulated in white blood cell differentiation. And of course, the difficult part would always be to establish their potential functions and mechanisms, but some of our preliminary data indicates that one of these candidates may influence the outcome of this white blood cell differentiation process. And naturally, any findings from this project will help to further our understanding of white blood cell physiology, the homeostatic conditions, and more importantly, white blood cells are implicated in a variety of diseases, not just those that are directly related to the immune system. So perhaps we could gain insights from the perspective of RNA splicing into why these cells do not function as required in these situations, and therapeutics that could help to alleviate these conditions. And that's the end of my presentation, and thank you for taking your time out of the day to listen.